Okay guys, so today I'm gonna introduce and explain to you DNA replication. So basically DNA replication has 4 steps. Step 1 is unzipping into 2 single strand, like here. Step 2 is primer binding. Step 3 is elongation, and step 4 is termination. And particularly for step 1, we have an enzyme called helicase, and it will unzip the DNA strands. Yeah, it will unzip into 2 single strands. And the DNA helicase disrupt the hydrogen bonding between base pairs, which will separate the strands into a Y shape, like as you guys see. This is known as the replication fork, and this area will be the template for replication to begin. Next, we have the second step, which is primer binding. So once the DNA strain have been separated, a short piece of RNA called the primer bind to the 3 end of the strain, and the primer always binds as the starting point of the replication. The primers are generated by the enzyme DNA primase. So basically the primer is the starting point for the DNA polymerase, which is in step 3. We have an enzyme called DNA polymerase, which is responsible for creating the new strain by a process called elongation. Here, this will come in, and the DNA polymerase is a builder. Here, so it will help matching these adenine cytosines, guanine, and thymine in order to build a new DNA strain to replicate. And for this strain, the DNA polymerase, which is moving in one direction, like here, and it will keep build until to the end to create a new strain of RNA. And however, for this strain, it will have a little different from this one, because so for this strain is known as the lagging strain. Why? Because the DNA polymerase will start from here, and we will go all the way to here. It helps. And the DNA polymerase will go back. Will actually go back here. So we have a new starting point. So it's known as the lagging strain. It is not go in one direction. It's moved from here to here, and then it will go back to here, and then just build, and then go back until it create a new single strain, a new DNA strain. So for the fourth step, which is termination, is known as once both the continuous and discontinuous strain are formed. The discontinuous particularly is the lagging strain, and the continuous is the leading strain. So an enzyme called exonuclease removes all the RNA primers from the original strain. These primers are then replaced with an appropriate base. Another enzyme called DNA ligase joins the Okazaki fragment, which is here. If the DNA polymerase from the lagging strain build from here. Then this fragment that they just built, known as the Okazaki fragments, and the DNA ligase will have glued them together. And we will bring the replication process to the end. And once the DNA in the cell is replicated, the cell can divide into two cells. Each will has an identical copy of the original DNA.